Bismillahirrahmanirrahim. Now it's time to revise our first lecture, Physiology of Hormone Secretion, in which we will discuss the synthesis and degradation. And detailed lecture is a separate lecture, but in this lecture we are just revising our first lecture. So quickly go through all the points which we have discussed in the physiology of endocrine hormone. The first thing regarding the physiology of endocrine hormone is the objective. Hormone synthesis is our first objective, then communication pathway, stimulus of hormone secretion, interaction of hormone, regulation of hormone secretion, regulation of hormone receptors, how they are regulated, and the clearance of hormone. And these 7 objectives should be discussed in this lecture. So what should mean by the endocrinology? It is simply the homeostatic function of hormone. So endocrinology define the function, that is homeostatic function of hormone. What are hormone? They are chemical substances secreted by the endocrine gland and distributed throughout the body via blood. So hormones, proper definition, 5 points. Chemical substance secreted in small amount from endocrine gland directly to the bloodstream in response to stimulus to cause physiological response as a target tissue. These 5 points should be mentioned in the proper definition of hormone. Chemical substance secreted in small amount from endocrine gland directly to the bloodstream in response to stimulus to cause physiological response at target tissue. Another way you can define hormone as a chemical substance released by a group of cells to control the function of other type of cells. Their effect may be limited to one target cell. Their effect may be different types of cell. For example, thyroxine, growth hormone, insulin. Growth hormone is not limited to only one cell. Growth hormone can affect our throughout our body. For example, they increase the size of bone where there is bone present. They can affect our muscle. So growth hormone, which is related to our growth from childhood to adult, so they can affect the different type of cell. In similar way, insulin is not limited to one cell. They can affect in hypoglycemic function in different type of cell, for example adipose tissue, muscles, so inside the liver. So their effect, their target cells may be different. So but some hormones are specific in their target. They are ACTH and estrogen, there is a good example. Because the specificity is defined by the receptor, not related to the hormone. Where there is a receptor, there should be a response. Now let's come to the types of hormones. 3 types, mainly lipid and water soluble, and steroid hormones, which are lipid soluble, amines, and the peptide hormones. And we become to memorize the amine hormones, there is a DONT mnemonic for this. So DONT represent dopamine, adrenaline, noradrenaline, thyroxine. VIP, represent the mnemonic for the peptide hormone, our most of hormone which are peptide nature. So vasopressin, insulin, parathormone, calcitonin, oxytocin, melanocyte releasing hormone, so adrenocorticotropic hormone. These are peptide nature hormone. Now discuss the synthesis. The steroid hormone synthesis by cholesterol, and the amino hormones, they are derived from tyrosine amino acid that is secreted by the thyroid gland. So the most hormone which are peptide in nature, they are only a short chain of amino acid. They are synthesized protein first, which is pre-pro hormone. Let's discuss the hormonal synthesis, which in these by the help of this illustration. The synthesis of peptide. The synthesis in this way: the DNA which is present inside this is your cell, here is your nucleus. In the nucleus, here is the DNA in the chromosome for the synthesis of a special hormone, peptide hormone. DNA convert into mRNA by the transcription. mRNA come to this ribosome, where what will do ribosomes convert this mRNA into pre-pro hormone by the way of translation. That is similar to the protein synthesis. So pre-pro hormone is our first protein, and then pre-pro hormone come to the endoplasmic reticulum, where they converted to pro hormone. So and the pro hormone come to Golgi bodies. After the Golgi bodies, the hormone is secreted, is final the shape of hormone is generated, and which will be stored in form of vesicle. So that is the that explains this mechanism. DNA in the nucleus convert to mRNA, mRNA come to ribosome convert it into pre-pro hormone converted to pro hormone by the endoplasmic reticulum, and pro hormone finally converted to hormone by the way Golgi bodies, and they stored in form of vesicle. That is synthesis pathway of peptide steroid hormone. The most important thing you should know that they are precursor of cholesterol, which is 27 carbon molecule. So they are when they are stimulated by the ACTH, which is a master gland hormone, when their positive stimulation come, they may be converted to testosterone, cortisol, DHEA, progesterone, estrogen. The biosynthesis pathway of all these hormone synthesis, we will discuss the adrenal gland discussion. So here just you remember their precursor of cholesterol stimulated by ACTH, and the hormones are cortisol, progesterone, estrogen, testosterone. So the mainly sex hormones are related to the steroid hormone. So these are synthesized by the cholesterol. What about the amines? We have already know that D represent dopamine, epinephrine, norepinephrine, adrenaline or noradrenaline, same thing, and thyroxine. These four are main our amine. So amine, how T3 and T4 thyroxine hormones are synthesized by the help of iodine. When iodine is incorporated to tyrosine, they convert it into T3 and T4, and the complete pathway will be discussed in the thyroid gland discussion. And tyrosine converted into dopamine, and which will further convert into epinephrine. It's not a simple by the help of enzyme for each step conversion. For dopamine, almost 2 enzymes are used for dopamine to norepinephrine, and epinephrine to finally norepinephrine to epinephrine. So the basic concept, the difference between norepinephrine and epinephrine is methyl difference. So that is the mechanism. Now coming to next, communication pathway. Communication pathway, different way of communication, as a lot of friends, when they each one has different way to communicate with other friends. So here is same mechanism. Every one has different communication pathway. For example, some have exocrine pathway. Exocrine, in which here is a acini cell, there is a secretory cell, and the exocrine, they communicate via duct. So here is the duct, the secretion from the cell, and via the duct the secretion is here, and it will affect the target cell. So the good example for the exocrine are saliva, pancreatic juice, so which are discussed in GAT module. So endocrine, how they affect, here is the secretory cell, here is a target. How they reach the target? The secretion, which is hormone. In case of endocrine, hormone came out of the secretory cell and from the glands. So the hormone come into the blood. Here is a blood vessel, through blood vessel distributed to their target, where the hormone came out of blood and reaches to their target receptor, where they perform their action. It may be through intracellular, may be extracellular. So good example, the whole module is endocrine, all the enzyme, the master gland, pituitary gland, the insulin, thyroid, So posterior pituitary are neuroendocrine, but all other glands mostly are in endocrine in nature. So the paracrine simply affect the neighbor cells which is present next to it. For example, histamine do what? They affect the parietal cell, Jasmine secrete the HCl. So they simply affect their neighbor cell. So there is no blood vessel, no duct, simply secretion came out, affect the target. From secretion came from this cell. This is a paracrine cell where their paracrine is secreted and affect the target. For example, if it's a parietal cell, the example for paracrine are histamine, somatostatin. Neural in which the secretion came out of neuron and affect the other target. So in this way you can see here is a synaptic cleft formation in which the acetylcholine is releasing from the axon terminal and coming to the and dendrites, or it may be a target tissue, it may be a muscle or any target tissue. That way of communication is neural. So the good example for this is our acetylcholine, epinephrine, norepinephrine, which autonomic neurotransmitter are the also same thing, neural or neurotransmitter communication. And other is which is combination of neural as well as endocrine. What does it mean by this? Secretion came from neuron, but the transport way is similar to endocrine. For example, the hormones came from this neuron, came out here and move through the blood and reaches to their target and affect there. So the posterior pituitary hormone do that function. For example, the hypothalamus, the oxytocin and ADH, which are synthesized in the hypothalamus, stored in the posterior pituitary, and when there is a stimulus come, the oxytocin and ADH are secreted out into the blood and reach to their target. So that way of communication is neuroendocrine. And other way of communication is autocrine. Secretion came out and into the interstitial fluid and affect back itself. In this way you can see here is the secretory cell, hormone came out, affect itself. A good example are interferon, which affect itself. In similar way, a hypothalamus, when there is negative feedback, they affect itself. So that way of communication is autocrine. Cytokine, they're similar to endocrine. Their effect may be a paracrine, exocrine, endocrine or autocrine, but they are simple short term, because all the endocrine, they are long term regulation, they are only short term regulation. Cytokine, the good example for cytokines are interleukin. And now discuss the stimulus of hormone, how the when should be a hormone secreted, when should be not. So their stimulus may be a humoral, may be a neural, may be a hormonal. In humoral, the concentration of ions is itself stimulus for secretion. For example, when there is high glucose in blood, it is a stimulus for insulin secretion. So, and similar, when there is low calcium level in the blood, it is a secretion for parathormone. So insulin for glucose and calcium, low glucose in blood is the humoral stimulus for the parathormone. Now look at the neural, in which nerve fiber give the stimulus to secrete the hormone. For example, the epinephrine, norepinephrine affect the adrenal medulla, which are secreted by the adrenal medulla when there is a stimulus, nerve fiber come here. So that way is the neural stimulus. In for example, the sympathetic, parasympathetic, and the autonomic nervous system regulation. So that is by the neural stimulus. And the hormonal, in hormonal, the hormone itself cause the secretion of other hormone, master gland. How they give stimulus to other hormone, because anterior pituitary, which are master gland, they affect the other hormone. For example, FSH, LH, ACTH, thyroid, FSH effect, the follicle luteinizing hormone related to ovaries, and ACTH which affect the adrenal cortex, thyroxine which affect the flat, simply they are which are tropic in nature. Tropic gland are they are stimulus for the secretion of other hormone, so that is stimulus of hormone, that is hormonal nature. Let's come to the interaction, how hormone interact with each other. There may be a synergistic effect, there may be antagonistic, permissive effect. So the in synergistic effect, the something is more than additive. We simply define that the if one hormone is affecting and other one is affecting alone, when they come at affect same thing, their effect will be more than their additive. For example, if one hormone is affecting, their result is 1, another hormone affecting, their result is 1. If they do their work collectively, so their result should be 1 plus 1 is equal to 2, but it is something more than additive, so their result should be 2.5, 3, 4, 5, not 2, because it is more than additive. So they result in the good example are hyperglycemic. When glucagon effect the alone, it is a very short hyperglycemic effect. Epinephrine, cortisol effect alone, there is a different effect. But when epinephrine and the glucagon or the cortisol effect collectively, their effect is much more hyperglycemia occurs. So that is the synergistic effect. Antagonistic, opposite effect, one increase, other decrease. Insulin decrease glucose level in blood, glucagon increase glucose level in blood, so that is antagonistic. In similar way, PTH and calcitonin, PTH parathormone increase calcium level in blood and calcitonin decrease calcium level in the blood, so that is opposite effect, that is antagonistic effect. In permissive effect, similar, very simple. The permissive mean permission, one hormone permit other hormone to do their effect. For example, cortisol which give permission to affect glucagon, for example in metabolism in gluconeogenesis. If cortisol is absent, glucagon itself cannot do their work. If the cortisol is absent, catecholamine cannot do their effect. So in similar way, thyroxine affect the growth hormone. Thyroxine is absent, growth hormone cannot do their effect. So in case of when there is thyroxine absent, the result is dwarfism. But dwarfism is not related to thyroxine, it is related to growth hormone. But growth hormone is unable to perform their action in the absence of thyroxine or thyroid hormone. So that is the permissive effect, which is very high yielding in the post graduation and in very national board exam. It is very important to discuss. Cortisol effect allow the glucagon to jump into gluconeogenesis. It allow the catecholamine to do their unvascular activity. So unvascular activity is related to catecholamine. In similar way, thyroxine allow the growth hormone to perform their action. For example, bone maturation, muscle strength, or the whole development process of a human or any other animal. So that is regulated by growth hormone. So let's come to the regulation of hormone secretion. Regulation of hormone secretion. Regulation of hormone secretion are related to peptide, because the steroid hormones are not stored. They are simply synthesized, released, and finally degrade. But in case of peptide which are in stored nature, so they can their secretion could be regulated in way of negative feedback or positive feedback. The negative feedback mean we are giving someone to not to do their work. For example, if you have attended a session, after a session someone came to you and say, man, give a feedback to this session. If you give negative feedback, it will be there will be if lot of negative feedback come, finally the session will be cancelled. In similar way, if you give positive feedback, more and more session will be discussed. So that is positive feedback and negative feedback. In similar way, if the hormone giving negative feedback to their stimulus, so their secretion should be stopped. In similar way, if they giving positive feedback, their secretion continue. And sometimes it is explosive in case of LH surge. Explosive means there is lot of oxytocin come in case of dilation of cervix. In positive feedback, in case similar in LH surge, lot of LH is increasing, self-promoting. That is positive feedback. It may be a autocrine. So negative feedback, it is simple. It may be a complex. In simple, glucose is high, it stimulates insulin decrease down the glucose. When there is glucose is decreased, there is no stimulus for insulin, so insulin also stop down. So that is negative feedback, simple. But there is a complex in which, look at this, the hypothalamus gives stimulus to anterior pituitary, and anterior pituitary, which is a master gland, affect the other hormone, because the tropic hormone affect the other. For example, thyroid affect the TSH affect the thyroid gland, ACTH affect the adrenal. So in this example, you can see affecting the adrenal causes testosterone secretion. So three important loop you should be aware. If the target organ is giving negative feedback, that is long loop. For example, testosterone giving negative feedback to hypothalamus, as well as if anterior pituitary, that is related to long loop mechanism of negative feedback. What is short loop? Hypothalamus affecting anterior pituitary, but anterior pituitary giving negative feedback to hypothalamus, that is short loop. And other way is ultra short loop. What does it mean by ultra short loop? Hypothalamus itself giving negative feedback to secrete their secretion, that is the ultra short loop, or by the autocrine pathway. So, and the positive feedback, in which we are giving stimulus to do more, yes man, come on, do that, that is the positive feedback. LH surge, oxytocin secretion by the dilation of cervix. What will happen in case of delivery? The cervix dilated, it gives stimulus to hypothalamus to secrete oxytocin by the posterior pituitary. Oxytocin causes uterine contraction, dilation of cervix. Already dilation of cervix causing the oxytocin increase, which will further increase the oxytocin secretion. So in this way, the dilation of cervix is more and more by the secretion of oxytocin more and more. So that is positive feedback. Let's discuss the receptor regulation. Receptor regulation is related to steroid, because the steroid are not stored, so their receptor should be regulated. By down regulation, if there is a lot of hormone is present, I want to decrease the effect, so I should decrease the receptor. What way of decreasing a receptor? Three way: decrease the synthesis, break down more, and inactive the receptor. For example, 100 receptor present, I want only 50 receptor for active, should be active. So I will inactivate 50 or degrade 50 or only synthesize 50, because I want only the effect should be desensitization or down regulation. So synthesize decrease, degradation more, inactivation. The example is progesterone. Progesterone downregulate itself as well as estrogen, because decreasing the receptor of estrogen as well as the receptor of mean the progesterone receptor. So upregulation, opposite effect. If there is a limited hormone secreted, what should I do? They increase the receptor. How can I increase the upregulation? Synthesize more, degrade less, activate more. Synthesis more and degrade less, because I already want to effect should be more. Sensitization. So that way is upregulation. Synthesis more, degradation less, activation more. So the good example is prolactin. Prolactin in case of breast, it increases its own receptor for the milk feeding. So in estrogen effect itself as well as LH receptor. So it is the upregulation. LH surge simply. And then another concept is here, it's not limit to upregulation, downregulation limit to itself. It may affect the receptor of other hormone. You can see here progesterone affecting itself as well as estrogen. In upregulation, you can see here estrogen affecting its own receptor as well as LH receptor. So that is the another concept. Finally, coming to clearance of hormones. Clearance of hormones is defined by two factors mainly: how much the hormone is synthesized, and now it's time to clear. In clear two way. One is to inactive, mean we should bound it, convert them into bound form, and other way is to remove it. Removal way by the liver, by the kidney, in urine, in similar way by the tissue degradation or tissue metabolism, by the liver, by the kidney, in urine, by the tissue, and are inactivated by the bound, converting them into bound form. That is related to the clearance of hormone.